Hello, everyone, and welcome to today's webinar titled Bridging the Gap, GenScript Solutions for Advancing CGT Immuno-Oncology Research and Therapeutics. This webinar is a part of the 11th annual event in the Cancer Research and Immuno-Oncology Week virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by GenScript. For more information about GenScript, please go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You will also notice that the speaker has provided a survey located on the right of your screen. Please take a few moments to answer the survey questions. It will be available during both the live and on-demand period. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the event. All right, now let's get started. I'd like to now present today's speaker, Daksha Patel, Senior Segment Marketing Manager, Cell and Gene Therapy at GenScript. For a complete biography on our speaker, please find it on the left of your screen. Daksha, you may now begin your presentation.
Good afternoon, everyone, and welcome to my presentation. Today, I'm going to give you a brief overview how GenScript has been at the forefront of the bridging the gap between the scientific innovation and supporting the discovery and development of cell and gene therapy, including the delivery of novel tools for cell engineering. Before we begin the presentation, I would like to start by sharing some background information about the company. GenScript is a biotech company that provides the life science service and the product worldwide. The company was founded in 2002 and its headquarters is in Piscataway, New Jersey, with additional location in China, Europe, and Japan. Key milestone of the company success are shown here. In 2002, GenScript launched the first custom gene synthesis platform. And 2013 launched subsidiary company Best Enzyme, which specializes in synthetic biology product. 2014, GenScript launched Legend Biotech, which is a subsidiary company specialized in cell therapy. And 2019, Legend Biotech GenScript launched the ProBio, which is a biologic CDMO service provider division. And 2022, Legend Biotech received the US FDA approval for CAR-T therapy. In summary, GenScript has the history of innovation and excellence in the life science. It is a prominent player in the field and known for its scientific innovation and contribution to the advancement of cell and gene therapy. Here is the preview of topic we will discuss today. I will begin with the introduction to the cell and gene therapy, and next we will focus on various stages of cell therapy development and investigate the type of tools needed for each stage and their importance. Let's begin with an overview of the cell therapy. Personalized medicine has made great progress in cell and gene therapy. Both the cell and gene therapy have a similar goal of treating, preventing, or curing the disease. However, they use different technique. GenScript can be providing the solution for both the cell and gene therapy. And gene therapy can be done in two ways. In vivo, which means that the therapeutic gene is directly delivered to the cell inside the patient body. And ex vivo refer to the process in which therapeutic gene is inserted into the cell outside the body prior to their introduction into the body. An example of this approach include the CAR T NK cell therapy, gamma delta T cell therapy, stem cell therapy, where the cells are engineered before used for the treatment. And cell therapy can utilize different type of cells, including the stem cells, immune cells, specialized cells from various tissue or organs. And cell therapy can be in general defined as autologous or allogenic. An autologous therapy uses the patient's own cell to treat the disease, while the allogenic therapy cell, in which cells coming from the single source and used to treat many patients. Here represent the diagrammatic representation showing the process of CAR T or CAR NK cell therapy, which involve the stage such as the isolation, activation, and cell engineering. Different CAR generation are shown into the figure for T cell therapy. Each CAR contains the structure that extends through the cell membrane, with one part located externally, another is internally. Typically, the extracellular segment of the CAR is composed of synthetic antibody fragment known as domain, which influence the receptor and ability to recognize and bind the tumor cell antigen. Additionally, newer CAR construct may incorporate alternative compounds such as the nanobody, ligand, protein, or scFv, single chain variable fragment for target recognition. Internal segment of each CAR contain the signaling and co-stimulatory domain that transmit the signal into the cell after the antigen interaction to the target. And the specific domain used can significantly influence the overall function of the cell. Beside the modification of the CAR, more advanced CAR immune cell therapeutic are being tested, which utilize precise insertion of gene to avoid the graft versus host disease. The process of cell and gene therapy involved the three main stages: discovery, development, and production. At the discovery stage, scientists aim to identify the disease target and evaluate the cell type that could address the medical condition along with the therapeutic effect. Subsequently, in the lead generation stage, optimizing CAR design and developing the cell engineering technique are crucial to enhance both effectiveness and cytotoxicity. Preclinical studies are then conducted using more complex animal model to evaluate the therapy safety, efficacy, and potential side effect. Finally, numerous cell therapy can progress to clinical trial where their safety and efficacy can be tested in human patient. If clinical trials are successful, the therapy may eventually receive regulatory approval and become a mainstream treatment option for various disease and medical conditions. Overall, CAR T-cell therapy is a complex and repetitive procedure that combines the molecular biology, immunology, translational research, bioinformatics, preclinical testing, clinical study to develop safe, more effective, and personalized medicine treatment. After obtaining the brief overview of the fundamental of cell therapy, this section of the presentation will focus on the solution offered by GenScript for research in the discovery stage, including the target discovery, lead generation, and CAR optimization. Presenting here is a comprehensive workflow for the discovery stage, which include the target identification and CAR generation. The target discovery phase is a complex process where researchers try to understand disease mechanism, identify the target gene, and analyze their influence on disease progression. Target identification can be approached by using different tools, beginning with the patient sample and using the sequencing technique and bioinformatic tools, potential target candidates are selected for in-depth investigation. Subsequently, leads are developed based on the target affinity ranking, which can also involve humanization if necessary. Once the target is identified, the lead identification and optimization stage, researchers focus on refining the candidate to qualify them as a potential drug. This involves optimizing entities like CARs or CRISPR-Cas system to enhance the specificity, efficacy through in-depth in vitro screening and functional assay. Throughout this process, various types of reagent and service are essential for gene expression, protein expression, target identification, cell and gene protein analysis, antibody development, functional screening, genetic engineering, and car optimization. GenScript offers a wide range of product and service for discovery work, and the next few slides will introduce different tools and their application. At the target discovery stage, analysis of individual cell provide information relevant to the human health and disease. For that, the gene screening and the gene sequencing are required. NGS-based molecular identification help to detect the mutation and variation in genomic DNA occurring within the tumor and their microenvironment. It also helps to identify the T cell receptor and help to understand the patient genetic information and can useful in diagnosis, treatment, and monitoring. Next generation sequencing workflow contains the three basic steps: library preparation, sequencing, and data analysis. And NGS technology workflow and various applications required synthetic oligonucleotide. And the quality of NGS oligo directly impact the efficiency and effectiveness of each workflow steps. For example, slightly impure oligo used in producing NGS library can significantly increase the sample cross-contamination while preparing the NGS library. And additionally, non-uniform capture probe library can lead to suboptimal sequencing during the targeted sequencing and potentially causing the loss of critical subsequent sequencing data for the low frequency genetic variant. Oligonucleotide also used in oligopool synthesis. Oligopool consists of the collection of multiple oligonucleotide which is used together for various applications, including the various library construction. For example, NGS target enrichment library, CRISPR gRNA library, and mutant library preparation. This library help in clarifying and understanding various aspects of disease mechanism and genetic association, like identification of genetic target or the marker. NGS target enrichment library help to detect the mutation and in genomic DNA occurring within the tumor. It also help to identification of T cell receptor. CRISPR guide gRNA library are ideal for high throughput screening of molecular targets, and it's also helped to understand the efficiency and specificity of CRISPR gene editing. Mutant library help in protein and antibody discovery and engineering, and it's allow complete control of mutation ratio and create near 100% coverage of desired variant. Incorporating the mutant library into the CAR T cell development and screening can expedite the CAR optimization process and enhancing the therapeutic potential and minimizing the potential adverse effect. In summary, oligo play crucial role in target discovery and can be used into the library preparation, amplification, clustering, sequencing, and custom application. They serve as a indispensable tool for producing precise and high quality data. With over 20 years of experience, GenScript offers high quality oligo suitable for all biological application. And GenScript proprietary electrochemical oligosynthesis platform enable the precise and cost-effective synthesis of user-defined sequence on chip. Additionally, our oligo pool synthesis platform allow high throughput synthesis of oligo, offering rapid and cost-effective approach to discovering the disease target. This platform provide not only standard unmodified oligo, but also allow us to synthesize complex oligo, which require the multiple modification. And we able to synthesize DNA oligo and RNA oligo ranging from milligram to gram level. To have successful sgRNA library or CRISPR-guided RNA library, the quality of gRNA in the pool is important. And it's also required control over the whole synthesis process. To construct the gRNA library, gRNA are precisely synthesized using our advanced semiconductor technology as a single standard oligonucleotide. And following PCR amplification, double standard gRNA are cloned into vector of choice to create screening library and typically a lentil vector used for efficient transduction. For designing optimized gRNA, GenScript offer online gRNA designer tool. And how CRISPR gRNA library used for hit identification also shown here, in which library are amplified back and transduced into the host cell to generate the mutant cell line, and the mutant can then be screened under the specific condition, such as by positive or negative selection to identify important gene into the pathway. And GenScript offer variety of CRISPR gRNA library, including the customized gRNA library, knockout library, and pathway-specific library. The use of CRISPR technology for target validation has a potential application and becoming increasingly common in various fields of research, ranging from basic research to drug discovery. And CRISPR allow reliable way to modify, manipulate gene for gain or loss function study and facilitating the understanding of target gene function in biology and disease. Here is a general overview of the how the ready-to-use GenScript kit or similar CRISPR kit can be used for target validation. The ready-to-use GenCRISPR kit simplifies the Cas-mediated gene editing. It supports every step of the gene editing workflow, including from sgRNA target selection, sgRNA synthesis, evaluating the efficiency of sgRNA, gene editing, and mutation efficiency detection. CRISPR technology finds widespread application in various domains, and for that it requires various CRISPR tools. GenScript offers the wide range of reagent and service that can be used for CRISPR-Cas technology in discovery application. This reagent and service are designed to help researcher in the various aspects of CRISPR-based experiment. And here, some of the reagents and service that GenScript provides are mentioned, which include the gRNA design and analysis tool, sgRNA synthesis, Cas9 protein, CRISPR plasmid and vector, CRISPR library, gene knockout service, etc. In cell therapy, neoantigens are very useful biomarker to distinguish cancer cell from the normal cell. And as the neoantigens are specific to the tumor cell, they are safe targets when used into tumor immunotherapy. New antigen peptide library is indeed useful for various purposes such as a functional screening and identifying important binding peptide, as well as TCR therapy, immunogenicity screening, and post-clinical testing. For example, T-cell truncated library is valuable tool for identifying specific epitope. Once the epitope identified, this sequence can be used to engineer or specialize T-cell for T-cell therapy. GenScript offered the wide range of catalog peptide product and peptide synthesis service, including the custom peptide synthesis, peptide library synthesis, peptide modification, and QC testing service. And different purity of peptide required for diverse application. So to support this, GenScript developed the two standardized peptide library. One is the standard peptide library, which guaranteed high purity and which is ideal for comprehensive screening as well as proteomic application, and microscale peptide library, which is suitable for preliminary screen. We also provide the complementary online tool for peptide library design. Molecular biology is an essential for understanding genomic disease mechanism and ultimately helping in the development of innovation and effective therapies. In the field of cell and gene therapy, molecular biology service find extensive application, supporting the study of DNA, RNA, protein, and other significant molecule, as well as their involvement in the cellular process like replication, transcription, translation, and gene expression. GenScript offer the range of service including the gene synthesis, cloning and mutagenesis, plasmid DNA preparation, vector construction, and gene to antibody development service. And this technique and tools play a very important role in advancing the progress of cell-based therapy. The extracellular domain of the CAR contain monoclonal antibody derived through the single chain variable fragment, and that determine whether the CAR T can specifically recognize the tumor cell, and it's play an important role in efficacy and safety of CAR T cell therapy. GenScript provide the diversified scFv development platform covering the entire process from antigen design and preparation, animal immunization to obtaining the scFv molecule and helping the customer to obtain their proprietary scFv sequencing. We also provide the mouse monoclonal antibody development service for generating high quality monoclonal antibody against any target of interest. A few slides earlier, I discussed the concept of precision mutant library derived from oligopool. And this library contains the genetically engineered version of the specific gene or protein, and with each variant carry an exact mutation within its sequence. Moreover, the platform includes the custom codon optimization capability and allowing researcher not only to optimize the expression for specific host organism, but also to incorporate the desired codon while eliminating undesired one with their mutant library. And this platform accelerates the scFv or sDAb or CAR generation process and reduce the downstream expenses. It does not require the extensive prior knowledge about the target antibody, which can save the time and effort during the screening and characterization phases. Precision mutant library can significantly contribute to the screening process of CAR development in the cell therapy. For example, CAR design include the various components such as the antigen binding domain, transmembrane domain, co-stimulatory domain, signaling domain. PML can be used to systematically explore the effect of different domain on the CAR therapy. And I presented the systematic diagram which shows how the precision mutant library can be used to antibody screening. Recombinant protein expression service play a very important role in the field of cell and gene therapy, and this service gives the therapeutic protein an antibody that not only enhance the CAR specificity and effectiveness, but also elevate the efficacy of cell and gene-based treatment. In the context of recombinant antibody expression in mammalian cell, challenge often include the low yield and long project timeline and the budget constraint. GenScript Turbo Cho platform addressed this issue by offering the high yield in the less time, supporting the antibody production at various scale. And platform can also use for various target, including the recombinant protein, bispecific antibody, FAB generation, VHH, or other fragment generation. And here is represented different type of turbochop platform offered by GenScript. Workflow for common CAR T cell therapy development shown here. And CAR T use the T cell engineering with CARs, which is usually SCFE or single domain antibody for cancer therapy. GenScript subsidiary company ProBio provide the CAR lead discovery, and key service highlights include in that discovery is the antibody discovery strategy, which is specially designed for CAR lead generation using the SPR technology to ensure the high affinity leads. And we have a well-established antibody optimization platform, and validation of CAR T cell function data is also provided. The validation is not focusing on the SCF itself, but it's also covered the functionality of CAR T cell. An in vitro bioassay for providing to validate the CAR T cell function, and specificity testing data also provided. Additionally, we can also offer the bispecific antibody discovery and lead generation services. Traditionally, CAR T cell therapy may incorporate elements derived from the mice, which can potentially trigger an immune response in human recipient and contribute to the graft versus host disease. However, the humanized version minimize or eliminate the murine component, reducing the likelihood of an immune reaction in CAR T cell therapy, and consequently tends to reduce the risk of graft versus host disease. GenScript also provide the high quality, fully humanized generation, human antibody generation service in addition to CAR lead discovery service. And for that, ProBio has established the partnership with the 3 transgenic animal platform, and through the integration of the single B cell screening platform with the humanized mice platform, GenScript ProBio can generate fully humanized antibody in as little as 1 month with to support your research. In cell and gene therapy, plasmid have various applications, including their use in research and preclinical study as gene delivery vehicle or gene editing tools, gene expression control, and it can also use in engineering of CAR T cell. Their versatility and capacity to introduce genetic modification make them essential tool in developing and advancing innovative therapeutic strategy. As the gene and cell therapy advance, there is an increasing demand for high quality plasmid DNA, and the requirement for making the plasmid DNA vary depending on its application. And high quality quality plasmid DNA manufacturing strategy shown here, and it combined with the gene synthesis, custom cloning to plasmid construction. In summary, plasmids are essential component in cell and gene therapy, and GenScript plasmid DNA preparation service accommodate a variety of clients, including small research lab and large-scale biotech and pharmaceutical company, ensuring the delivery of high-quality plasmid DNA with 100% accuracy in the full insert sequence. GenScript provides the stage-appropriate plasmid service from research grade to preclinical. And if customer wants to construct the plasmid by themselves, we can also provide the DNA assembly kit for plasmid construction. If customer choose to perform the plasmid construction themselves, we also offer the MAC Quattro plasmid purification system, which is a fully automated solution designed for purified of large scale and high quality plasmid. And the comprehensive solution consists of a system controller that provide simultaneously operation of 4 independent purification module, and MAC Quattro powered by magnetic bead technology. And the key benefit of the system include high throughput up to 24 sample screening, flexible culture volume ranging from 50 to 200 ml, and ready to use consumable available for quick use. Cellular assay, also known as a stable assay, um, and serve as a valuable tools in biomedical research and drug discovery screening. It's supporting us for efficient quantification of cytotoxicity, biological activity, biochemical mechanism, and off-target interaction study. In the cell therapy, cellular assay is a crucial step in evaluating the efficiency of CAR toxicity and efficient, and it can also use in vitro research. This assay offer the distinct advantage as they generate the complex and biologically relevant data, providing a more physiologically meaningful assessment. And GenScript offers stable cell line development service, including the assay cell line development and CRISPR cell line development for protein expression and cell line engineering study. In the field of cell and gene therapy, efficiently generated CRISPR knockout knock-in cell line is an easy and efficient tool to which can use in immunotherapy target discovery, drug screening, and study functional research study. And CRISPR edited cell line models are created using the CRISPR-Cas9 technology to knock out gene and knock in target sequence, resulting in genetically edited cell line capable of stable passage. Schematic of process used to generate the knockout cell pool shown here, and steps included in this process is like design and synthesis of gRNA, transfect the cell, analyze the editing and knockout efficiency, and conduct the assay. The generation of CRISPR-mediated gene knock-in or knockout cell line involves substantial workload, and it is and thoughtful design of gRNA template is necessary for generating functional gene knockout and knock-in, which enhancing the editing efficiency. GenScript offered the diverse CRISPR gene editing cell line, and we utilize chemically synthesized long single stranded gRNA, which give high editing efficiency and minimum toxicity through the RNP delivery system. We also support the multiple delivery methods, including the lentivirus and plasmid. And GenScript also provide customized knockout cell line service for multiple gene and customized knockout cell pool service. GenScript SSL line service workflow and overview shown here. It is also a multi-step process from vector construction to cell line delivery. And cell-powered stable cell line are ideal for cellular assays such as the target screening and cytotoxicity assay. Additionally, we also offer the single-chain derived cell line featuring the Fc receptor, G protein receptor, and immune checkpoint cell line. And this cell line are helpful for cell-based bioassay and allowing the assessment of binding affinity between the antibody proteins and immunotherapeutic targets. So to support the discovery work, including the target discovery, target identification, protein expression, gene expression, cell protein analysis or antibody discovery, or cell engineering, GenScript offers a wide range of product and service, and all are listed here. After getting the brief overview of cell therapy target discovery and lead generation, this section will now shift our focus on the tools which is offered for cell engineering and the manufacturing of cell therapy. Common cell and ex vivo gene therapy workflow shown here. Cell therapy process has three key steps: cell isolation, activation, and engineering. And producing cell and gene therapy is a complex process. It involves the multiple products from different sources, making them more complicated. And sourcing the necessary material to produce the consistent product in one of the most significant manufacturing challenging for most biotech company. In addition to preventing the GMP blockage, choosing the right reagent supplier or partner who can provide specialized reagent regularly over long period with the desired quality and on-time delivery is crucial. Any disturbance in the supply chain for essential product or consumable can cause significant delay in production of life-saving drug. GenScript offer top-notch reagent, RUO to GMP quality, and supply chain support to overcome this hurdle. For cell isolation, GenScript offer the cytosine 1000 instrument, which is a highly efficient and automated equipment and easy to operate, and has key features like barcode scanning, customizable programming, real-time workflow monitoring, and full traceability. It can be easily integrated with other instruments and allowing a smooth workflow for preclinical and clinical development. Cytosine cell separation and activation kit include the antigen-specific magnetic nanobead, and this can be used for fresh or frozen PBMC or leukophoresis. And these reagents are sterilized, non-toxic, and available in RUO and GMP grade. Recombinant protein, including the cytokines and growth factors, are necessary ancillary material which is utilized during the manufacturing into the cell therapy process. Example, in CAR T cell therapy, T cell require the growth factor to expand, which are provided by the medium and serum. And one of the growth factor is IL-2, which is an interleukin-2, which binding to the T cell receptor and promote the polyclonal expansion of T cell. For cell therapy research, GenScript offer more than 2,000 ready-to-use cytokines and growth factor. Even if these recombinant proteins are not presented in the final product, but their quality can impact the stability, safety, potency, and purity of the product. And GenScript now provide the new GMP platform to convert all the RUO grade protein to GMP grade, and they are suitable for use in preclinical and clinical stage. And this can help a smooth transition of your project from research grade to GMP. Cell engineering is a crucial component of the cell therapy process, and using the various engineering strategies, researchers aim to enhance the safety, effectiveness, and versatility of the CAR T cell therapy. CAR have a modular design with the 4 major components, and each component has a distinct function, and optimal molecular composition of the CAR can be achieved through variation of these each constituent protein domain. Toxicity and efficacy associated with the CAR T cell therapy can be addressed using the engineering strategy. In addition, CAR gene construct can be further modified to engineer CAR T cell with armor protein expression, which typically enhance the T cell function or favorably modify the tumor microenvironment. Variation of each component of CAR construct enable the fine-tuning the functionality and anti-tumor activity of resultant CAR T cell product. CAR with various designs are being developed to improve the safety and efficiency of this therapy across the various cancer. Lately, researchers have been actively refining CAR design through the incorporating synthetic biology principle. This concept have been um, integrated into the immune cell, leading to the improved safety and effectiveness in cellular immunotherapy. And schematic diagram of non-viral CAR T generation shown here, where gene editing technology like CRISPR-Cas9, mRNA-Cas9 peptide have been used by researchers to engineer complex biological system with unique functionality and which helping the new era of precision medicine. Advancement in mRNA through electroporation or nanoparticle also offering the promising opportunity into the T cell engineering with the mRNA. The selection between the viral and non-viral CRISPR cell engineering method depend on the specific research or therapeutic goal. Research carefully evaluate this factor to determine the most suitable method for the need. Lentil viral vector established the current standard for delivering the transgene payload into the manufacturing of CAR T cell, a well-developed method supporting the high expansion level. However, this method may be associated with excessive expression and consequently activation induce the cell death or T cell exhaustion. On the other hand, non-viral approaches are precise, cost-effective, and accelerate the cell therapeutic development. However, they may have certain drawbacks such as the lower insertion efficiency and limited availability of GMP resources for this work. Due to the limitation of viral-based CRISPR delivery, non-viral approach becoming the more popular in immuno-oncology therapeutic research. Non-viral method use CRISPR, mRNA protein, and synthetic guide RNA to deliver the component, and there is no need for additional process like transcription and translation. As a result, non-viral method provide better control over the over the viral method. CRISPR-based gene editing application require Cas9 enzyme, which act like the molecular scissor. And to support this work, GenScript offered the GMP grade gene CRISPR Ultra Cas9, which reduced the off-target by over tenfold and while maintaining the robust on-target efficiency. Researchers have recently developed the Cas fusion protein, which is known as a CRISPR-off, and that can modify and silence the gene without the editing the underlying genome. And the epigenetic memory persists throughout the cell differentiation, and it's naturally passes through the future cell generation. And gene activity can be reactivated using the different fusion protein called CRISPR-on. To support all this work, GenScript offered the customized CRISPR protein service also. Combining the sgRNA with Cas9 protein into the ribonucleoprotein RNP complex offer the additional advantage over the traditional plasmid or virus-based delivery system. Unlike the traditional plasmid or lentivirus delivery method, CRISPR RNP delivery is intact complex and do not require cellular expression. And synthetic sgRNA is a single oligonucleotide which available is ready to use form, and it eliminates the need for cloning and sequencing. And it's also a best option for complementary DNA-free gene editing when combining with the Cas9 protein. In addition, in recent study, it was found that sgRNA with 5-triphosphate modification product through in vitro transcription can induce the prompt inherent immune response and lead to the cytotoxicity in human and human cell. However, chemically synthesized sgRNA without this 5-triphosphate modification demonstrated much better editing efficiency in the cell, and thus the supporting the chemically synthesized sgRNA are the most ideal reagent for CRISPR gene editing. And GenScript provide the two different type of sgRNA synthesis. One is the easy edit sgRNA, and the another one is a safe edit sgRNA. One powerful application of CRISPR-Cas genome editing technology involves the precisely inserting DNA sequence via HDR pathway, and selecting the suitable HDR template is critical. And GenScript now offers the high-quality sequence-verified HDR template in both linear and circular format, which is GenX single standard DNA, double standard DNA, and GenCircle double standard DNA. And it can be used in application like as knock-in template, as a viral packaging plasmid, as well as gene of interest or non-viral vector. Sorry, we have to repeat this slide. So can you edit them?
Yes.
Hello. Yeah, I'm restarting them. One powerful application of CRISPR-Cas genome editing technology involves precisely inserting DNA sequence via HDR pathway, and selecting the suitable HDR template is critical. GenScript now offers the high-quality sequence-verified HDR template both in linear and circular format. GenExact ssDNA support the lowest cytotoxicity for high efficiency and minimize the off-target effect, which is ideal for T-cell therapy. GenVen ssDNA is covalently close and linear DNA, which enhance the stability, and it can be used for large-scale screening and scale-up in cell and gene therapy. And GenCircular dsDNA, which is a mini vector with no antibiotic resistance gene and can increase the knock-in efficiency up to 30% and shows the lower cytotoxicity, which is the ideal replacement of plasmid and HDR template. In this experiment, cell modification with the CRISPR gene editing tools is simplified by using the RNP complex combined with the synthetic HDR template and electroporated into the isolated and activated T cell. And diagram illustrates the precise integration of knock-in T cell using the RNP approach through a non-viral protocol. And key step of the process include primary human T cell isolated from blood and stimulated before the electroporation. An HDR template were first added, and then the RNP complex consisting of sgRNA and Cas9 protein were added. And after incubation at the room temperature, cells suspended into the buffer were and mixed with the HDR template and RNP by pipetting. And after the electroporation, engineered T cell can be readily expanded for downstream measurement. In this case study, Dr. Brian from UCSF developed the hybrid ssDNA with Cas9 target site as a novel payload for non-viral method to genome editing. Diagram of hybrid single standard DNA with Cas9 target shown into the figure. And Dr. Brian collaborated with GenScript, uh, and GenScript provided single standard DNA, and they were able to perform therapeutic gene editing on primary T cell at clinical scale using the GenScript single standard DNA. To support the non-viral CRISPR cell engineering work, all CRISPR reagent services are available in the stage-appropriate format to support the R&D work as well as the tech development and production work. To fulfill the high-quality specification, we can provide IND declaration support and the GMP production for all CRISPR's reagent. In recent time, the potential of mRNA has gained increasing recognition for the development of innovative biotherapeutics. Scientists predicting its application in various fields like immunotherapy, gene editing, regenerative medicine, and vaccination, simplify self-remanufacturing and cell cycle independent performance, and risk-free mutagenesis increase the mRNA adaptability into the therapeutic field. And figure shows the structural element of IVT mRNA, and each of them has a specific role. And each of these elements can be optimized and modified in order to modulate the stability, translation capacity, and immune stimulatory profile of mRNA. From the last few years, growing number of researchers exploring the IVT mRNA for development, testing, and application of CAR T cell-based immunotherapy. mRNA can use for both reprogramming of T cell as well as gene editing with the CRISPR-Cas family of nucleus, as represented here. And it can also be used in ex vivo gene editing, such as with sorry, I have to repeat this slide. From the last few years, growing number of researchers exploring the transcribed IVT mRNA for the development, testing, and application of T cell-based immunotherapeutic. mRNA can be used for both reprogramming of T cell as well as gene editing with the CRISPR-Cas9 family of nucleus. As shown in the figure A, reprogramming of T cell using the mRNA create the CAR antigen that target specific or tumor related antigens are emerging as a safe therapeutic approach. And it can also be used for ex vivo gene editing, such as with the CRISPR family. And this technology offers improved precision of gene editing as shown in the figure B. An mRNA mediated delivery of nucleus has several advantages, such as the no genomic interference, a potentially low off-target rate, and high editing efficiency. And mRNA leaves no genetic residue in the recipient cell and offer much safer CAR therapy than the viral CAR. Therefore, mRNA has become a versatile tool in molecular biology and biotechnology, and high-quality modified mRNA required for different process. Before sourcing the high-quality modified mRNA on small or large scale, crucial factors like plasmid DNA, mRNA integrity, quality of the poly A tail, proper mRNA capping, and uridine modification, and efficient lipid nanoparticle packaging, minimization of impurity should be taken into consideration for achieving the success. All these factors affect the end product quality, yield, expression efficiency, translational efficiency, stability, and toxicity. mRNA synthesis is a multi-step process as shown into the figure, and to avoid the risk associated with the engaging multiple suppliers for different service, developers are generally seeking for full service solution that cover gene synthesis to the linearization, IVT mRNA manufacturing, and quality control. And GenScript recognized this need and has developed an integrated manufacturing workflow which covers the wide range of service and QC analysis. And our workflow include the codon optimization, gene synthesis, plasmid preparation, mRNA synthesis, lipid nanoparticle formulation, and ensuring the production of high optimized mRNA with excellent quality and expression efficiency. We also providing off-shelf mRNA to meet your research requirement. Nowadays, neoantigen activated T cell therapy is gaining popularity, and neoantigen activated T cell therapy offering a personalized, efficient, and safe treatment approach. The general process of NRT cell manufacturing presented here, in which first tumor cells are and normal cells are isolated from the patient, and the neoantigen in the tumor cells are identified by sequencing, and the design and synthesis of neoantigen peptide is required. After that, neoantigen presenting cell generated by transfection or electroporation of neoantigen to the normal cell like DC or B cell, and then neoantigen loaded cell co-cultured with the PBMC derived T cell, and later on neoantigen reactive T cells are isolated and undergo expansion, and after that product can be infused back to the patient. And here also shows the represented the basic procedure of neoantigen reactive T cell immunotherapy, where tumor T cell and CD8 T cells are first isolated from the patient. Then the neoantigen in the tumor cell are identified by sequencing, and the neoantigen presenting cells are co-cultured with CD8 plus T cell, and neoantigen reactive T cell are shorted, and after in vitro modification and expansion, the neoantigen reactive T cell product can be infused back into the patient. Additionally, neoantigen specific TCR T cell therapy shows promise for AML treatment, and that shows encouraging anti-leukemia effect. In comparison to CAR-T technology, neoantigen activated T cell therapy is a highly personalized treatment, and it can work better for a wide range of people and is more effective and has fewer problems. And scientists are still studying and learning more about it. And regardless of therapeutic avenue, one commonly linking all form of neoantigen treatment is the requirement for reliable neoantigen peptide, as they are the only reagent required for therapeutic discovery, development, and efficacy screening. Peptide synthesis for neoantigen is an important part of the cell therapy process. However, neoantigen peptides are commonly quite difficult to be synthesized, as they can be highly hydrophobic, very significantly in length and charge, and have strong tendency to aggregate. Therefore, new antigen peptide synthesis can be significantly bottleneck into the timeline of clinician generating personalized immunotherapy. And in order to help new antigen community, GenScript has launched their neoantigen peptide synthesis platform with NeoPrep algorithm, which identifies synthetic difficulty and recommend the best synthetic technology based on each individual peptide intrasonic characteristic property. And GenScript has been providing reliable custom peptide synthesis service from 14 years, from last 18 years, with our proprietary microwave assisted prep power peptide synthesis platform. And GenScript is now able to offer high quality peptides in the GMP or preclinical grade, and custom peptide synthesis package option cover the fast peptide synthesis, rush peptide synthesis, regular peptide synthesis. Many researchers using the viral vector in cell and gene therapy for cell engineering for delivery of functional gene to replace defective gene or to cure the genetic disorder. Our optimized vector and third generation lentil viral production platform offer the researcher reliable and efficient gene delivery tools. We offer the lentil virus packaging with higher titer and quality to ensure the downstream safety, including the QC testing. In addition to lentil virus, we also offer AV packaging service. And this vector can be used for knockout screen cell engineering and therapeutic gene delivery. And GenScript ProBio is end-to-end CDMO service provider for CGT application, which also providing the customized option for making the plasmid as well as the lentil viral vector and AAV vector. So uh, GenScript has a capability to providing the research grade as well as the clinical grade viral vector as well as the plasmid service of the different scale. So in summary, for cell and gene therapy, we are offering the various service and product. Most importantly, we offer the GMP grade material and customizable option for different stage of research and clinical development. Overall, GenScript aim is to provide researcher and industry with the tools and service which needed for developing and delivering safe and effective gene and cell therapy. And GenScript Life Science offer the wide range of service and product to support not only the cell and gene therapy, but also to support the antibody drug discovery as well as the vaccine solution. Thank you, everyone, for attending this presentation.
Thank you, Daksha, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. All right, let's get started. Our first question asks, could you provide me with additional information on CytoSync 1000 automated cell isolation?
Yes, so Cytosine 1000 automated cell isolation is designed to automate the process of isolating specific cell population from complex sample, and this technology aims to improve the efficiency, accuracy, and reproducibility of cell isolation, which is an essential step in many research and clinical application. And GenScript Cytosine 1000, it's easy to use, highly efficient. For it can be used for isolation as well as depletion, and the technology which use in this instrument, it's like immunomagnetic nanoparticle which selectively isolate the targeted cell population of interest, and it's allowed to isolate full leukopak of half leukopak, and within the 2 hours you can able to finish complete leukopak isolation. And the key features of this technology, it's like the whole instrument allow the main control is easily accessible while the process is ongoing, and barcode scanning for easy reagent entry available, as well as it's allow the lot tracking and file export, ensuring the full traceability. This instrument also allow the real-time monitoring through the workflow screen, so you can see the real-time overview of the process, and you estimated completion time also you can understand. And it also offers the customizable protocol which allow the user to optimize the isolation process for their specific need, and which may be the very unique features which we're offering, so customizable reprogramming. And if you're interested to know more about this Cytosine 1000, definitely connect to us. We will provide some technical data which we collected, how they perform with the different start sample, and we can share that information to you.
Great, thank you. It looks like we have time for one more question here, so we'll wrap up after this one. How does GenScript ensure the quality and reliability of its genetic tools and reagents?
Well, this is a good question. And GenScript follow the quality management system in line with international standard like ISO 9001 and covering all stages of product development, manufacturing, and distribution. And the key quality assurance steps which we always taking care of, it's like purity and integrity check, quality assurance throughout the production, validation and testing, and managing sure that we have the quality standard complies with regulatory requirement to provide the best quality reagent and service to the customer. And for that, we GenScript also have like advanced manufacturing technology. So during they follow like strict manufacturing process and utilize the advanced technology to produce the high quality product. Uh, and regularly audit and inspection is carried out throughout the production house. And we have experienced scientific team which focus on this quality control, and they apply the um, very hard QC testing method to assess the performance, purity, and consistency of genetic tools and reagent.
Well, thank you again, Daksha Patel, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until October 7th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.